Today's essential question, which is number 3 out of 4, is still dealing with the topic evidence for evolution. So, this essential question, still dealing with evidence for evolution, is specifically answering the question: how do molecular sequences and biogeography provide evidence for evolution? How do molecular sequences and biogeography provide evidence for evolution? Your first left hand column for today: molecular sequences. Molecular sequences. Molecular sequencing, which refers to DNA or protein and amino acid sequences, can be used to analyze molecules of life in groups of organisms. At the molecular nucleotide or amino acid level, organisms from bacteria to pigs have many similarities. Now, all living things carry their genetic code in either DNA or RNA. There are also molecules that pass on traits to offspring in every living organism. Many genes and proteins are also homologous across the living world. For example, one particular protein, cytochrome C, which functions in the process of cellular respiration, is very similar in all living cells. In another example, bacteria and eukaryotes are very different kinds of organisms. Yet, they both share the same genes that code for the ribosomal units used in protein synthesis, which suggests that bacteria and eukaryotes shared a common ancestor a long time ago. A very long time ago. By comparing sequences in the proteins or DNA of different organisms, scientists can determine how closely related they are. Generally speaking, the greater the number of differences, the greater the amount of time that has elapsed since they had a common ancestor. Molecular sequencing is just another piece of evidence that supports Darwin's original idea that modification through natural selection, life evolved from a common ancestor. Now remember, natural selection is the process by which traits or alleles become more or less frequent in a population, depending on the advantage or disadvantage that they confer on the survival and reproduction of the organism. New alleles arise in a population due to mutation. If a new allele confers a reproductive advantage, it will increase in frequency. As such allele changes accumulate, new species may arise. New left-hand column, molecular clocks. Molecular clocks. Molecular clocks are techniques that use genetic mutation rates to determine historical time when divergence of species occurs. There is some evidence that some evolutionary changes take place at a molecular level in a clock-like fashion. Over millions of years, random mutations build up in any given stretch of DNA at a fairly constant rate. This makes changes in DNA a powerful tool for analyzing and evaluating the evolutionary histories of related organisms, which enables scientists to calculate the dates when evolutionary lineages split. For example, imagine that a length of DNA that is found in two species differ by 2 bases. And we know that this entire length of DNA changes at a rate of about 1 base per 10 million years. That means that the two DNA versions differ by 20 million years of evolution. Since each lineage experienced its own evolution, the two species must have descended from a common ancestor that lived at least 10 million years ago. So when comparing different organisms, the more genes that are compared, the more accurate the dating.
Evolution results from changes in DNA. These changes are caused by random mutations, changes in the DNA sequence. Mutations are often detrimental, but some mutations are neutral. Neutral mutations neither benefit nor decrease an organism's ability to survive and reproduce. Neutral mutations are evolutionary changes and occur in a clock-like fashion. They act as a sort of molecular clock. Molecular clock is a technique used in evolutionary biology that estimates the age divergence of organisms from a common ancestor by looking at differences in their DNA. Scientists measure the differences between the DNA in certain genes. Often, mitochondrial DNA of organisms is used because its rate of mutation is well established. They use this molecular clock to determine when organisms diverged from a common ancestor. In this example, two different species had a common ancestor sometime in the past. To determine when they diverged from a common ancestor, scientists use a molecular clock based on a gene that they know changes by 2 bases every 25 million years. They observe that the species differ in the DNA of this molecular clock gene by 4 bases. If the species differ by 4 bases, they must have diverged from a common ancestor around 50 million years ago. It's difficult to calibrate the molecular clock against actual time because the rate of mutation depends on factors such as generation time and population size. The calibration leads to some uncertainty in dates of divergence. However, using multiple genes along with fossil data can help narrow down the dates. Scientists have identified genes that have more constant rates of change than others. These make the best molecular clocks. Biologists have used the molecular clock technique to determine the date of the divergence of humans and chimpanzees. The data show that the last common ancestor of chimps and humans lived around 5 to 7 million years ago. This ancestor evolved into various hominid species, one of which is an ancestor of modern man. In 2008, researchers found DNA in Neanderthal fossils from this cave in Croatia. The scientists compared the mitochondrial DNA of humans and Neanderthals to estimate divergence of our modern lineages from these ancestors. Molecular clocks are an imprecise but useful tool. The technique has been used to investigate important issues such as the origin of modern humans.
Your next left-hand column, biogeography. Biogeography. Biogeography is the study of the distribution of organisms, species, ecosystems, and biomes all around the world. Factors affecting the distribution of organisms in biological communities include habitat area, climate and soil composition, longitude, latitude and elevation, and the geological history of a region. Please take down that list. Factors affecting the distribution of organisms in biological communities include habitat area, climate and soil composition, longitude, latitude and elevation, and the geological history of a region. In Darwin's time, there were many aspects about the distribution of organisms that could not be explained. For example, why were the ostrich-like birds in Africa, South America, and Australia so similar? These are continents that are thousands of kilometers apart. Why did so many remote islands have species that were only found on those islands? Why did these animals and plants, or how did these animals and plants get to these places? Darwin used his ideas about evolution to explain some of these observations. Darwin considered biogeographic distributions as evidence for evolution and natural selection. On his visit to the remote Galapagos Islands, Darwin noticed the presence of many unique species. He realized that island-dwelling plants and animals were often geographically isolated from other islands or the nearest mainland. Because of this geographic isolation, island organisms could breed only with other island organisms. As time passed, island plants and animals often evolved into new species that were distinctly different from the mainland species from which they descended. This is the reason islands often have many endemic species, which means like a species of plant or animal that are found nowhere else. For example, the Galapagos Islands were the home of a number of species of finches. These birds had many similarities, but all were slightly different. They all occupied different niches. One scientific example is that these similarities were a result of the finches evolving from a few common ancestors that managed to find their way to the islands long ago. Over time, the finches adapted to vacant niches, and this is an example of adaptive radiation. Better understanding about the movement of the continents in Earth's past has helped to unravel some of the mysteries of plant and animal distributions. For example, scientists know that the continents of Africa, Australia, and South America were once joined. This probably enabled the common ancestor of today's ostrich-like birds to colonize all three, now widely separated, continents.
Adaptive radiation is an evolutionary phenomenon in which a group of organisms rapidly evolves into many new species to take advantage of new ecological niches when they become available. This may occur after the formation of new islands by tectonic movements, upward growth of coral reefs, or the eruption of a volcano. It also occurs most dramatically after a mass extinction event such as that of the dinosaurs millions of years ago. Other life forms evolved through adaptive radiation to take their place.
Adaptive radiation is when a group of living things either survives a mass extinction event or colonizes an environment that there are no competitors in. Because of that, because there's no competition, either the competition is wiped out or there was no competition to begin with, lots of variations can survive. And so in a geologically short period of time, what was once just one survivor, one species, becomes many, many different survivors with different ways of life.
As a result, one species will give rise to many others as they evolve to fill newly available niches. In Hawaii, at least 54 different species of honeycreeper birds have evolved. They all descended from a single species of finch that arrived in Hawaii more than 4 million years ago, when the volcanic islands were still very young and there were many new habitats to colonize. The honeycreepers diversified rapidly to take advantage of the new niches. Some evolved long, curved bills to extract nectar from Hawaii's lobelia flowers. Others developed short, stout bills for cracking open seeds, and some developed hooked bills to probe tree bark looking for insects. Another example occurs in Africa. The Great Rift Valley region is home to 15 lakes and many species of fish belonging to a family called the cichlids. Studies have found that species of cichlids isolated by the formation of a new lake quickly undergo adaptive radiation to form many new species. In a few million years, close to 2,000 species of cichlids have evolved. Each of these is unique because it is adapted to a specific niche. As new lakes and new niches become available, new species of cichlid fish evolve. There are numerous examples of organisms that have evolved through adaptive radiation to fill newly vacant niches, whether it's the lemurs isolated on the island of Madagascar or the anole lizards of the Caribbean islands.
At this point, you should be able to generate an answer to today's essential question. How do molecular sequences in biogeography provide evidence for evolution? Go ahead and stop the video at this point and write your answer to today's essential question in the summary box at the bottom of your notes.